Now we are talking about how the information that is kind of coming to the ears are going into the brain and kind of getting processed inside the brain. There is a pathway that we receive information from this pathway, the information that I told you, which is decomposed to frequencies and intensity. And also we discussed that there is a time factor here, and there could be even a time differences in terms of the right ear or left ear that we are receiving. So we have frequency, intensity, time. These information are going to be coded and go into the brain. We have different kind of stations that this information is being processed and going to the final area that is called auditory cortex. Auditory cortex is located in a part of the cortex that we call temporal cortex, okay? This is the temporal cortex. The temporal cortex, as we discussed before, has one part that we have superior temporal gyrus, we have middle temporal gyrus, and inferior temporal gyrus. We discussed that we have different gyri. But there is another gyri which is sort of transverse here. We call that transverse temporal gyrus. Okay? That is called the auditory cortex. But this transverse temporal cortex could be divided in different pieces. I will discuss about different pieces that we have in transverse temporal gyrus. But this transverse temporal gyrus is located in a sulci that we call Sylvian sulci. Sylvian sulcus. So this one is the Sylvian sulcus. And this one is basically the temporal, the transverse temporal in the Sylvian sulcus. Okay? Would you just join me numbering the numbers of synapses that we have? We have one synapse here. The second synapse could be happening here. 2. We can have another synapse here. We can have another synapse 4 here, and then to the cortex. And these two pieces are from thalamus, right? We have discussed about that. We have discussed about inferior colliculus. This one is midbrain. These are what we call cochlear nuclei, which are located, as you can see here. These are in the kind of medulla part of the brain. So these pieces are working together. I'm not going to go to the details about them, even talking about what is the role of reticular formation and lateral lemniscus and other pieces. It's just too much detail. I mean, what we are interested is that the information is going to the brain in different synaptic level and then going to thalamus and from thalamus to the cortex. Okay, that is what we know about the auditory process. There are definitely details about each of these kind of levels of information processing. What we need to know is that when we talk about that transverse temporal gyrus that we had here in both sides, this transverse temporal gyrus is receiving information from both ears, both right and left. So each side would receive information from both sides. And there is a little bit difference compared to what we have with the visual system. So you can start to see what are the differences between the auditory and visual system. What we have in the transverse side, in the macaque, in the monkey, what we have here, that is basically that transverse side inside. We have a core, belt, and parabelt. Okay. In human subject, which is going to be a little bit more complex, we have what we call HG, then we have PT, and then TPT. What do they mean? This one is what we call Heschel gyrus or Heschel gyrus, and this one is called planum temporal, And this one is a temporoparietal cortex. It might be kind of still a little too much for the kind of the anatomy to learn, but I will give you several other kind of figures to give you a better sense about how these pieces are working together. But what we have here, as you can see here, within the temporal row, we have a transverse temporal, which is in a transverse kind of shape. And then in this transverse gyri, we have different gyri. So we have one gyri, which is called Heschel gyri, and then we have another gyri, which is called planum temporal. And they have different information processing roles. The core, which is here as you can see, the core, or the Heschel gyrus, is the primary visual auditory cortex. This one is primary auditory cortex. And these one are secondary auditory cortex. But when we go out of these environment, out of these blue parts, are not auditory only. These pieces have information from other sensory modalities as well. Whenever we are here in the blue part, whatever we have is just the auditory process. But when we go beyond the blue parts, other parts would have information from other sensory modalities. And I will tell you why this is important, to have primary auditory processing, secondary auditory processing, and then multimodal sensory processing. So we receive information from the ears to the thalamus. It is being relayed in thalamus in MGB, medial geniculate body, or medial geniculate nucleus. It is relayed to the temporal cortex. In the transverse temporal cortex, it starts from the primary or Heschel gyrus, and then from that it would go to the next step to the planum temporal, and then from planum temporal to the multimodal sensory processing. And in this process of moving from, let's say, auditory system up to this system, and then this system from kind of primary to the secondary, from primary to the secondary and to the multimodal, we start to understand what is happening in the sound in the environment around us. And that is how this system is working. But I will give you more details about how this system is working in the next steps. If you remember, we discussed about kind of the Brodmann areas. As here you can see different Brodmann areas. What we have here, this primary visual cortex, the auditory cortex, is Brodmann area 41, and then the secondary one are going to be Brodmann area 42. So just kind of having those kind of numbers in your mind. And then when we go to the area that is multimodal, that one is going to be number 20, so Brodmann area 22, which is doing a more kind of advanced combined sensory processing from both auditory and other information that we are receiving from the environment. Okay, I will discuss about that in the next steps. Just to give you a kind of level of sense in terms of Heschel gyrus, so this one is, the dictation is Heschel's gyrus. Here you can see this is the Heschel gyrus, and this one is the segmented. But what do we mean by segmented? Segmented means that the green parts are gray matter, the red ones are basically white matter. So that is the segment. It doesn't matter very much, but this one and this cut is, which cut is this? Is this coronal, sagittal, or transverse? You can write it on the chat space. This one, this cut. Yep, that is true. Good. So yes, this one is coronal. And when we have a coronal kind of cut, when we start a coronal cut, if we want to define the coronal cut, we report Y number. This one is a transverse, or the axial. And when I'm reporting an axial cut, I need to report its Z. Why? Because in transverse or axial cuts, I'm changing the Z over the time. I'm changing from lower Z's to higher Z's because I'm cutting in this direction. But when I'm cutting in coronal, I'm going from back of the head to the kind of the front of the head, and I'm cutting in the Y direction. That's why I need to report what is the Y. So as you can see here in this cut, you can see the facial gyrus in this cut, okay, minus 16. So it should be an area like here. So it is back to the to the head from the anterior commissure. If we go to the front, it would be positive Y. This one is a negative Y because in the back side of the head. So we are cutting from here, that you can see it here. And this is what I really kind of like you to develop in terms of having a 3D sense about the brain. And sometimes it makes it a little bit complex in terms of understanding these dimensions when you are kind of using these atlases. But you need to start to develop this skill because then you can go yourself inside those atlases and see what is happening. And that's the reason I'm asking you during last few sessions to do your homeworks and your exercises on working on different brain atlases. Because if you develop the skill to be able to understand these figures, then you can start to learn those things by your own. And that is exactly the qualification I want you to have after this class, to be able to understand these complex concepts by kind of studying the publication in the field without kind of really being dependent to others to process the information for you. Okay, this one is the planum temporal. Okay, this one is planum temporal. And it's quite interesting that the planum temporal on the left side is almost always larger than the planum temporal on the left side. And that's another level of question, why we have planum temporal in such a larger size on the left side, but on the right side it is kind of smaller. But the facial gyrus on the left and right is quite the same. So you can see here is the facial gyrus. So the facial gyrus in the left and right is quite the same, but the planum temporal would be different on left and right side. And I will discuss about why we have this kind of interesting differentiation. And I should say that the auditory system is pretty much bilateral. The language system is pretty much left-sided in most of the brains. And that's the reason that we have this part which is involved in language processing in a larger size. Okay. Another important thing that is important to kind of be considered in understanding what we are discussing about is that we discussed that in cochlea we have a tonotopy. And the reason that we have tonotopy is that we have wires or filaments or fibers in this kind of line, and these fibers are going from really small ones to really kind of longer ones. You have a kind of, even the musical instruments, when you have a wire, if you have shorter wires, if you kind of resonate them, if you vibrate them, you have a low pitch sound. But if you have in the higher frequency, or if you have kind of a longer wires, if you kind of vibrate them, you have high pitch sound or a kind of low pitch sound in a lower frequency. Okay. And that's the kind of there are some kind of physiological details about that. I'm not going to go to that. But we have a tonotopy here. Okay. And it seems that we have similar tonotopy in the primary auditory cortex in cat. So we have the same reflection in the cat. So the same map is being reflected to the cortex. And we have that kind of differentiation between different pieces of information, even on the cortical level. And if you remember, we have similar idea on what we call the retinotopy on the visual cortex, that we had the same spatial map of the surrounding environment that we have on retina, because on retina the place kind of principle is about the place of items in the external environment. But the place in the auditory system is about the frequency, because we decompose sounds to frequencies, and different frequencies are being perceived in different places That's the reason that here we have the tonotropy is frequency-based, but the retinotopy that we discuss in the visual system is the spatial-based. So that, I mean, we have some similarities and differences. And it might be interesting that in human subjects, this is the retinotopy on the primary auditory cortex. Here you can see that there is a sort of retinotopy here, tonotopy here, from low frequencies to higher frequencies. So that is being reflected to the auditory, to the primary auditory cortex. When we go to the secondary auditory cortex, and then we go to the other parts, that map is not being preserved completely. So the map is being distorted, or the map is being more, let's say, mixed together. So the information is not going to remain in the frequency basis when it goes to the other levels and layers of information processing. So we start from the tonotopic map in the simpler kind of areas that are processing the information, but when we go from that area to the next levels, it's just being mixed together, okay, over the time. And it also might be interesting that when we are reporting these maps, sometimes we report them in the individual level. So let's say this is the map of Hamid's brain. But sometimes we make an average, something like 20 subjects. We put 20 subjects together and make an average. And you can see that even there are variations between different individuals that could kind of make different variations in terms of we do not have the same maps in different individuals. We have individualized maps of these retinotopy and processing in the information on the cortex. That makes things sometimes really complex in human cognitive neuroscience because you do not have a single map. For each individual subject, sometimes you need to make a personalized map. Okay, so that is another part of the visual processing anatomy, and we discussed about, let's say, how this information is being kind of recorded in the cochlea and goes from the kind of bottom to up in what we call ascending pathways to the cortex. But interestingly, there are pathways from the cortex back to cochlea. We call them descending pathways. And these descending pathways, what is the role of these descending pathways? Why do we need descending pathways? So we'll discuss about that later. But it's interesting that these descending pathways would go up to the cochlea. And I mean, they have a role. So we'll discuss about that role in the next steps as well.